Bonjour tout le monde. Habituellement, c'est Audrey ou moi qui faisons la présentation de notre invité. Mais aujourd'hui, on va laisser M. Christian Messier, directeur de l'ISPAR, faire la présentation. Vas-y, Christian.
Merci, Philippe. Oui, ça me fait évidemment un très grand plaisir d'accueillir Julie Messier. Quel joli nom. Donc, ce n'est pas par hasard, hein, mais elle est, je vais le dire tout de suite, c'est ma nièce. J'en suis très fier. Je vais quand même commencer à vous dire un peu ce qu'elle fait dans la vie, mais elle est prof à Waterloo, en fait, depuis fin 2018. Donc, ça fait un peu plus d'un an déjà, presque 2 ans. Mais avant de parler de ça, je voulais vous dire quand même des choses un peu plus personnelles parce que très tôt dans sa vie, elle s'est intéressée aux arbres et elle grimpait dans les arbres. Hein. Très tôt, elle n'avait aucune peur de grimper dans les arbres. Plus tard, elle a développé plus un intérêt pour des parois rocheuses, les parois les plus abruptes. Et je pense que ça la caractérise assez bien. Elle n'a pas froid aux yeux et surtout, elle ne se laisse jamais abattre par les obstacles. Elle les grimpe tout simplement. Elle s'est beaucoup promenée dans sa carrière. Elle a étudié au Panama. Elle a fait des études au Panama pendant quelques mois, un été, je pense. Au Panama, elle était en France. Elle a fait son doctorat aux États-Unis, en Arizona. D'ailleurs, j'ai remarqué que l'Arizona, depuis le passage de Julie, est passé du rouge au bleu. Je crois qu'elle en est partiellement responsable. L'autre chose intéressante, c'est qu'elle a fait un bac à l'UQAM. Puis elle a fait aussi surtout un projet de fin de bac avec quelqu'un que j'aime beaucoup, Dan Nisha, où elle a même publié un article. Et elle a fait son doctorat en Arizona, mais aussi avec un autre, en co-direction avec une autre personne que j'apprécie beaucoup, Marty Lekowicz. Et vous avez vu par son sujet qu'elle travaille sur un sujet très chaud, hein, il est très fonctionnel. Une autre de ses caractéristiques, en regardant son CV, j'ai remarqué qu'elle avait des articles qui sont très cités et d'ailleurs quelques-uns déjà qui sont plus cités que mes meilleurs articles. Donc, c'est de bon augure, Julie. Et souvent, les gens vont dire à Julie, ben, ton nom, c'est Christian Messier, mais je pense de plus en plus, je vais me faire dire, ah, tu es l'oncle de Julie. C'est ça qui va arriver dans le futur. Donc, je vais terminer pour dire que je suis très, très fier de ma nièce et de Julie, et je te cède la parole.
Merci beaucoup, Christian, pour l'introduction. C'est très gentil, je suis très touchée. Moi, j'aime dire souvent quand les gens me disent « Ah, Christian, c'est ton nom », je dis « Oui, je suis la version 2.0 ».
Améliorée.
Différent. Donc, je vais quand même transférer en anglais, vu qu'en fait, j'ai fait toutes mes études en anglais, puis la terminologie, je la connais mieux. Donc, si je vous donne la présentation en français, elle va durer 1h30 plutôt que 45 minutes. Donc, today I decided to give you a broad overview of the research I've been doing throughout my career, instead of focusing on a single study. So the overarching theme is I'm exploring the limit of trait-based ecology, and I'm looking at the cause and consequences of phenotypic variation and integration across scales. So to set the stage a little bit, let's see if my pointer is working. Can you guys see the pointer? Yes, okay. Sorry, Zoom issues. Oh, there we go. So yeah, so ecology is the science that studies the relationship between organisms and the environment. And like any science, it tries to find generalities. So a goal, I would also argue that ecology is one of the hardest sciences in which to find generalities because we are looking at over 9 million species, each that has a unique set of genes and traits and a unique evolutionary history. So in ecology, finding generalities is not only a core goal, but it's also a core challenge. The classic approach to find generalities has been to study individually each species. So that's generally referred to as autoecology. However, an important problem with autoecology is that here we have my two field site. DCI is where I did my fieldwork for my master's. Mont-Saint-Hilaire is where I did my fieldwork for my PhD. So by studying individual species, if I find something about a species in BCI, I have no idea if it applies to other species in BCI, let alone other species on Mont Saint-Hilaire in the temperate forest. So today I'm exploring another approach. Instead of autoecology, it's called trait-based ecology. So instead of focusing on the species as the unit of study, we're focusing on the traits or the phenotypes as the unit of study. And the goal here is to find generalities, because the idea is if we can find rules that apply to specific phenotypic traits, then hopefully those rules apply across species in a given site, or even better, across different species across sites. So I'm going to be talking about functional traits. So I'll start by giving you a quick definition of functional traits. So they're defined as any measurable morphological, physiological, or phenological traits which indirectly impacts individual fitness. So this definition is actually couched in a very central paradigm of both ecology and evolution. And that's the idea that the phenotype of an organism determines its fitness. So then phenotypes are largely adaptive. So when I think of this subject, I like to show this figure, which is from a classic paper by Arnold in 1983. Here you see that the different traits Z and their covariance, as shown by the curved arrows linking them, affect different performance metrics, such as survival, growth, and annual seed output, for example. And together, these performance metrics then determine fitness, which is W. So example of traits that Z are, for example, here, leaf mass per area or leaf area. We can have wood density for wood, or rooting depth in roots. So trait-based ecology has been broadly adopted in the last 20 years because it makes three very important promises. The first one is what I started my presentation with, and it's the promise of generality. So to restate this, by measuring properties that are common to all plants, we can compare species from diverse environments and diverse taxa. And actually, the trait-based approach has really delivered well on that promise, because it has described ubiquitous trait correlation among species, which are typically referred to as trait dimensions. But just think of sets of traits that trade off together and that describe major axes of phenotypic variation. And one of the main successes of this, or the main successes of this, of defining trait dimension, is that then we can organize the global plant diversity along these different axes to understand their different plant strategies. My presentation and my work relies heavily on trait dimensions, so I'm going to take a little bit of time here to explain them. So in the literature, we have found 5 major trait dimensions. The first one is known as maximum plant height, then the seed size, then a set of leaf traits called the leaf economic spectrum, a set of wood traits called the wood spectrum, and Corner's rule, which is less broadly studied, but also very important because it describes the architecture of plants. So I spend the rest of the talk discussing the leaf economic spectrum, the wood spectrum, and Corner's rule. So here I'm going to explain those trait dimensions to you briefly. So here's a tree, and the first trait dimension is called the leaf economic spectrum, and it's called economic spectrum because it defines the rate of return on carbon investment over the leaf's lifespan. So on one end, you have leaves that have high rate of resource acquisition, but they don't last very long because they're poorly defended. And on the other end of that spectrum, you have leaves that have, they're good at conserving resources, but they have a very low rate of return or photosynthesis, so carbon acquisition per unit time. So some of the traits that are typically associated with that dimension are called maximum photosynthetic rate, leaf lifespan, and leaf mass per area. The second trait dimension is the wood spectrum, sometimes referred to wood economic spectrum, but I prefer not to call it economic spectrum because it has nothing to do with the rate of return of carbon or other nutrients. It was classically thought to be made up of a trade-off between 3 functions, but here I'm presenting a framework where, actually, which I believe is much more robust. It's been presented by Pratt and Jacobson in 2017. It's like a fantastic read, anybody that wants to learn more about wood traits. And it's arguing that really the wood spectrum, or any wood, has to trade off, find a way to optimize 4 different functions. So resource storage, and then the transport, which has the safety aspect of sap transport and the efficiency aspect, and mechanical support. So really any trait combinations that we see is a trade-off among these 4 functions. And I think that of the trait dimensions, this one is the least understood, probably because, again, it involves many functions. So some of the traits that are typically measured to reflect that dimension are wood density, vessel diameter, and trachea diameter, and modulus of elasticity, which is basically how much force do you have to put on the branch or on the stem to have it bend by a certain distance. The third dimension, which I really like, again, not studied very much, is called Corner's rule. It's really just an architectural dimension. It's actually fantastic because I think it's one of the dimensions that has found the broadest support in the literature in terms of it's been shown to hold true within trees, among oaks, or coexisting oaks in the different studies, or among coexisting plants, and also among different species in different biomes. So I think it's kind of the most ubiquitous of all trait dimensions that I've found in the literature. So those architectural relationships arise as the plant is trying to maximize mechanical support, light acquisition, and sap transport. So that leads to relationship among architectural traits, including the area of leaves, the diameter of branches, and the frequency of branching on those branches. Okay. The second important promise of the trait-based approach is to improve our ability to make predictions. And that, again, is almost, by definition, should be true if the trait-based approach is correct. First, with functional traits, they're defined as traits affecting fitness of individuals. So by definition, here I've highlighted in red that relationship between trait and fitness. If we're measuring the right trait, we should be measuring traits that affect fitness. So that's like self-evident. But also, we should be able to find relationship between traits and environmental gradient because, again, we make the assumption that phenotypes are largely adaptive, so that the correct traits or the traits that are best adapted to individual environments should be found there. And last, it makes the promise of integration across scales, meaning that if we can measure the same trait, namely, for example, leaf mass per area, at different levels, at the level of an individual organ, of an individual plant, of a population, of a species, or among, at the level of the community, it means that we can then, it gives us a common metric to link the different patterns and processes that are acting at each of those scales. So now leaf mass per area can inform the physiology, growth rate, demography, distribution, and assembly of plants. So because of all these promises, traits have become very popular in plant ecology since about late 1990s, early 2000s. And just to illustrate that, I had, like 5 years ago, I had looked for this little graph of the numbers of published paper with the word trait in the title in the field of ecology per year. And you can see here on the left-hand side, there's been a dramatic increase, more than tenfold over that time. And the citations per year for these papers has actually increased exponentially. So I like to make the analogy that basically ecologists have fallen in love with the trait-based approach. But like any love story, at the beginning, there's a honeymoon phase. And in the honeymoon phase, you only see the good sides. But after a little bit of time, you start realizing that your partner is not just perfect. So you start seeing the things that don't work so well. But in any good relationship, what you do is figure out a way to work through them and figure out where the boundaries should be, what compromises should be. So in that same way, when I started my PhD in 2010, I was looking for understanding what are the limits of the trait-based approach. So, okay, it makes this promise of generality. There's all this hype about it, but where does it fail us? And by understanding where it does and it doesn't work, we can then work to improve the approach where it needs improving. And also another important question is, are the underlying assumptions valid? So we need to test this. And interestingly, as I defended my PhD in 2015 in December, a few months before that, a paper came out in Ecologia by a few of the founding fathers of the trait-based approach, namely Bill Shipley and Peter Reich. And they are making basically the same argument that I'm going to be making throughout this talk, is that, yes, the trait-based approach is great, but we still have a lot of work to do to reinforce some loose foundations. So I'm not the only one kind of questioning the extent to which we should be relying on the trait-based approach. So my work is focusing on two frontiers. I like to think of them as two challenges. They're also two of the topics that I'm most excited about. And this is the ideas of scale. And when I say scale, I'm specifically thinking about biological scale or also organizational scale. So whether I'm measuring a trait at the level of the organ, individual, population, species, or community. And the other frontier is that of phenotypic complexity. So am I working with a few traits or all the traits? Now, clearly all the traits is never possible. So like how many traits do we need to work with to get a good picture of a plant's ecological strategy? So when I talk about complexity, I'm talking not only about that number of traits, but also those interactions among those traits. So those curved arrows between the Zs that we saw in Arnold's figure. So when I started my PhD, I would argue that the field of trait-based ecology is roughly here where my bubble is. And what people tend to do is, since we've defined these trait dimensions, so here's a doodle of trait dimensions where every circle is a trait. The lines connecting them shows their correlation. So here we have three sets of traits that are correlated within each other, but not among them. So that would be, for example, here in orange, the wood economic spectrum, in gray, Corner's rule, and in green, the leaf economic spectrum. So because we find these three trait dimensions, people assume that, that because they occur at global scales, they're understood to be ubiquitous, and they understand that I can measure a single trait from each of those trait dimensions to reflect that trade-off, and I can measure it across all of the scales. So here, this is why I have that arrow. So I'm gonna characterize an individual by measuring these three traits independently, and then I'm also gonna characterize a community by measuring these same three traits independently. However, this approach leads to, not approach, this method, or sampling method has two issues. The first one is that we know that the whole phenotype affect performance. So by measuring only three traits, not looking at their correlation, we're effectively making that assumption that these three traits are not correlated, and they are the three ones that determine the majority of variation in performance. The other issue with this is that trait dimensions are defined globally, and actually in my talk I'm going to make the case that a global trait dimension or a global pattern does not mean a ubiquitous trait pattern. It just means that it occurs at that scale. It doesn't mean that that's true all the time in all the contexts. So we have actually not yet really done all the work to show that these three trait dimensions, those correlation patterns, are also true if I'm looking at 2 or 3 different species in a community, or if I'm looking at many individuals within a population. And this is very important because my ability to measure leaf mass per area as an index of the leaf economic spectrum is based on the assumption that it's correlated with and only with other traits in the leaf economic spectrum. Here in this lower doodle, if my leaf mass per area is also correlated with resource conservation, but it's also correlated with, I don't know, wood density, then variation in LMA could mean a lot of different things, not necessarily variation in leaf economic strategy. Okay, so that's the setup for my work. In this talk, I'm gonna go over 3 questions that, in order to address them, I've had to work with different levels of phenotypic complexity and at different scales. So first, I'm going to go over the question, do global trait dimension actually hold at local scale, by presenting 2 studies that were done with the different sets of traits. Then I'm going to talk about what are the drivers of trait integration at local scale. So I'm going to present one study here that was done at comparing different populations, multiple traits among different populations. And then my third question, I'm going to ask, okay, are we actually measuring the traits that do predict plant performance well? So let's validate that assumption. So here again, I'm going to present 2 different studies, one at the population level, one at the individual level. Okay, so first, do global trait dimensions hold at local scale? So the assumption that people have been making in the field is that because it's global, it means it's ubiquitous. So here, trait dimensions are ubiquitous. So let's look at that. So in a paper that I published in 2017, here, that ecography paper, I basically discuss, based kind of in an armchair argument, like why we may not expect trait dimensions to hold across scales, across biological scales. So the argument is that we shouldn't expect the leaf economic spectrum to occur in local data sets because mainly the argument is basically based on a sampling effect. So at local scales, the range of trait values is reduced. So the noise surrounding the trait correlation can actually drown the signal of the leaf economic spectrum. So let me illustrate that because I am aware it's not easy to explain simply just with words. So this is a figure that comes from one of the first papers describing the leaf economic spectrum. It was published by Peter Reich in 1997. So on the x-axis, we have leaf mass per area on a log scale, I believe. On the y-axis, we have leaf nitrogen concentration, also on a log scale. And this study was done using a variety of species from a variety of biomes across the world. And we see that even with this log-log scale, and even using basically the range of growth form and of species across the world, the strength of a correlation coefficient is between 0.5 or 0.6, depending on how you do your analyses. However, so imagine that now I'm going to do a local study, and I'm going to look at the correlations between these two traits. So now here, because it's local, my range of variation on the x and on the y-axis is necessarily reduced. And here, so if I zoom on that figure, actually this isn't data from the same paper, this is my own data from BCI. This is what the data point looks like. So maybe the leaf economic spectrum is still there. So there could be part of their relationship, part of the variation in each trait that's driven by a correlation with the other trait. But really, that's what I mean by the noise about that signal. It's possible, it's large enough that it drowns the leaf economic spectrum signal. So in other words, what this means is that at local scale, it's possible that leaf mass per area responds to drivers of variation, say, for example, the amount of light, that are not the same drivers of variation that mainly affect the other traits. So here, leaf nitrogen concentration could be largely determined by differences in soil fertility at the kind of the site level. So then these two traits are affected by different drivers, so that create variation in one trait that's independent of the other, and that creates more noise about the signal, so I'm not able to detect the LES anymore. Okay, so in that same paper, I kind of explained my my argument, and then I also did a little lit review to say, okay, if this is the case, like what have we found so far in these local scale studies? Like, do we find a leaf economic spectrum? And I actually found that there's a lot of studies that address a different question using a leaf economic spectrum traits. So they don't specifically ask, can I see the leaf economic spectrum? But they do report their correlation coefficients. And they oftentimes we found that these trait relationship that we expect in the LES, they're either not there, so their correlations are not significant, or they're significant but in the opposite direction of the global patterns. So the literature does support that the leaf economic spectrum is not necessarily ubiquitous. And finally, I looked at my own data set. So that was, I used the data from my master's work in Panama. And I find that my different leaf economic spectrum traits, they do show moderate levels of correlation in the order of 0.4 to 0.6. And they also respond differently to different drivers of variation. So again, because the correlation coefficients are moderate, it gives the space for these two traits to respond differently to different drivers of variation. So in conclusion, from that paper, I'm arguing that global trait dimensions do not necessarily hold at local scale. So I want to emphasize that it's possible that they hold in local scales, but we shouldn't just take it for granted because it's possible that they don't. Okay, so to understand then how trait dimensions behave locally, my next question is, I wanted to ask is, okay, are these trait dimensions independent from one another, or are they kind of related to one another? So globally, we've studied these different trait dimensions. It's not really possible, or it's not easy to ask whether they're correlated together at the global scale, but I decided to do that. Like, are those trait dimensions really reflecting independent trade-off in a local data set? Because that's what I can do as a PhD student. So as I mentioned in my introduction, this question is important to answer because by measuring a single trait to reflect each dimension, we implicitly make the assumption that these dimensions are independent. So I wanted to test this. So I'm going to give you the overview of my sampling design for my PhD work and a lot of the other papers that I'm going to present in this presentation are actually building on this data set. So this is my field site, Mont Saint-Hilaire, which is like a half an hour east of Montreal as the crow flies. Yeah, and so on this mountain I measured 24 angiosperm tree species, which is the majority of the tree richness for angiosperms. I didn't measure gymnosperms because I know that there's a very strong signal of phylogeny between angiosperm and gymnosperm in the traits, and I wasn't interested in finding correlations that were mostly driven by that difference between gymnosperms and angiosperms. For those 24 tree species, I measured a total of 380 trees, and these are, I had 15 to 20 saplings per species, and when I say saplings, I mean trees with a size of 1 to 5 centimeters at breast height, diameter at breast height of the stem. In my study site, then I measured, for each tree, I measured 20 traits from all plant organs. So I had 7 leaf traits, 9 branch traits, 2 stem traits, and 2 root traits. Just for the sake of it, I'm going to show you that table. I'm not going to go over it, it's too long, but the other people that do trait-based ecology might find it informative. So those 20 traits were chosen to reflect 6 physiological functions, so architecture, mechanical support, conductive safety, conductive efficiency, resource acquisition, and resource conservation. And you may recognize these physiological functions as forming the 3 trait dimensions that I explained earlier. So I basically studied Korner's rule with the wood spectrum and the leaf economic spectrum. And so yeah, these traits are color-coded just to help you kind of visualize the results. So my postdoc advisor, Mark Vellen, always gives me feedback in my talks that I'm too conceptual and I don't show my study organisms enough. So here's the slide where I show you some of the field work so you can see the study organism. So this is me separating second and first order roots on fine roots. This is my field assistant Andréane measuring leaf area. This is a picture of a branch that we use to measure architectural traits. This is us extracting roots to try to put in the growth bag to get the fine roots on our saplings. This is Margarita Murphy at the University of Arizona doing wood volume to get stem wood density. This is my other field assistant, Emily, who was measuring tree height. And this is just a cross-section of the twigs that we stain to measure all our wood traits, or like vessel anatomy. So this is saffron and stain. I think these pictures are just so beautiful. I love showing them. And that's it. So back to conceptual stuff. So we would expect that if trait dimensions are independent, they should be correlated mostly within trait dimensions and not among trait dimensions. Now, I did a network analyses to address that question. And so I did PCAs and network analyses. I'm just going to show you the results of the network analyses. I think they're a lot more kind of intuitive and easy to follow. So to test whether my trait dimensions were independent, I did a modularity analysis. And what that does is that you define a priori those modules. So here the modules are the trait dimensions. They're synonymous. And it tests what's the ratio of correlations within the module relative to correlation between the modules. And then it compares that to a null model where the correlations were drawn at random with respect to those modules. So a value of 0 tells you the module doesn't exist because there's as many correlation among module as between module, which is within module as between module, as expected by chance. Okay, so what did we find? So this is actually a result. So here are the acronyms of all my traits. As a reminder, green traits are leaf economic spectrum traits. Orange traits are wood spectrum traits, and the gray traits are Korner's rule. And the modularity analysis is actually very, very almost exactly 0. So based on that definition of modules, based on those trait dimensions, there are no modules. I mean, the trait dimensions are definitely not independent from each other. So here, just to explain a bit more my figure, the red dashed lines are negative correlations. The solid blue lines are positive correlations. And the thickness of the line reflects the strength of the correlations. And I mean, just kind of looking at this, you can really see that the orange doesn't cluster together, the green doesn't completely cluster together. This group of leaf traits do, these don't. And then these architectural traits, like 3 of the 4 kind of do, but branching angle, which should cluster, doesn't cluster. So yeah, so no, they don't form modules. So the answer to this question, are trait dimensions independent at local scale? Well, definitely not in this study. So trait dimensions may not be independent or even present locally. Actually, yeah, so if you read the paper, there's support for the presence of Corner's rule in that data set, but the other 2 trait dimensions don't really exist in that data set, which kind of reinforces the previous study that I presented. Okay, so the conclusion for that first question is that global trait dimensions may not hold at local scales. And the implication of this is that the trait dimension framework may not be the best framework to use for trait-based ecology in more local scale studies. I think that maybe using the information used in trait correlation networks or correlation matrices might be more useful and for local scale studies. Again, I'm not saying that it's impossible that those global dimensions will hold locally. It's just that we shouldn't take it for granted. So if we're going to study a system, we first should check what the trait correlations look like in our system. And then if the leaf economic spectrum is there, then we can use just SLA or LMA to reflect that dimension. Okay, moving on to the next question. So, okay, so if traits are not integrated into trait dimensions locally, then what are those local drivers of trait integration? Yes, that's what I just said. If trait dimensions do not organize the phenotype, then what does? So to address that question, I borrowed an approach from quantitative genetics. And what I did is I compared the intraspecific correlations of different species. And this is I love that approach. I was excited to find it because what it does is that, I think I illustrate this, yeah, in the next thing. The similarities of the different species in their intraspecific correlation points us to ubiquitous intraspecific correlation that may reflect fundamental relationships. So, for example, if I have 2 traits, trait 1 and trait 2, here every color is a different species. So here in this diagram, all the intraspecific trait correlation of the different species would be falling on the same line, so they would be sharing that relationship. So I'm calling it a fundamental trait relationship. I'm not calling it a universal constraint because it's possible that those correlations that are present in all the species arise. It could be because there's some sort of constraints that act on all those species. It could be like physiological constraint or biomechanical constraint, but it's also could be that there's this pervasive natural selection that means that it's always adaptive for those traits to be correlated in all those species. So I have to be careful when interpreting that. So that's why I'm just going to stop at saying there's something fundamental going on here. So I can use those same results from comparing my intraspecific correlations of different species to then ask, okay, there's going to be differences, and can I use these differences to link them to gradients? So for example, again, illustrating this, if my 2 traits, the correlation of those, are not correlated the same way among all the species, then I can ask, okay, well, is the strength of a correlation associated with some environmental gradient? And here, as an example, maybe it's the correlation strength increases with the amount of light. So here I'd have no correlation at 20 par, slightly higher correlation at 100 par, more at 300, and like really strong correlation at 1000 par. So if I can draw these links between environmental gradients and differences in trait correlation, then I can say, ha, like this environmental variable is likely to be responsible for that trait association. Okay, so again, same data set, 24 trees, 15 to 20 sapling per species. I did not have a full data set for my root traits, so I dropped the root traits and I have 17 traits for this study. Just to be clear, when I talk about intraspecific correlation, I'm specifically referring to static correlation. So static correlation is defined in the evolutionary literature as correlations among individuals of the same developmental stage within a population. And this is important to specify because I'm not looking at correlations due to ontogeny, so different developmental stages. And I'm not looking at correlations among different individuals of different populations. So it's really kind of genetic differences among individuals that drive these correlations. Okay, so to continue with my, okay, so I see, I don't know if you've noticed, but here question 2.1. So are there fundamental trait-trait relationships? So this bigger question was, what are the drivers of trait integration? I can use similarities, and I can use differences to answer two different questions. And because the methods are actually different for each of those two sub-questions, I'm first going to go over the methods for the looking at similarities. What did I find? What does it mean? And then I'm going to go over looking at what are the differences, are they related to environmental gradients, and what does it mean? So 2.1, are there fundamental trait-trait relationships in that data set? So I first calculated the intraspecific trait correlation matrix for each species. So here's species A, traits 1 to 17 here, then you fill that with the correlation coefficients. And then for each pair of traits, I asked, are these traits correlated significantly in all species? I had to come up with a way to try to address this. So what I did is I'm going to show you the results in a visual fashion. I think it's the most effective. So here, imagine this, a pair of traits, trait 3 and trait 15. Now I have 24 species, so I'm going to have 24 intraspecific correlation coefficients. So here's a distribution of those correlation coefficients among my 24 species. But now I have 17 pairs of traits, so that's over 200 unique pair correlation, trait pairs. So I'm going to do that distribution for all of my trait pairs. So now I'm going to have a graph with a bunch of distribution. And what I'm going to ask is, are there some pairs of traits that are significantly correlated in all species or almost all species? So to do that, I built null model. I calculated this, the non-significance thresholds across my data set. And it was actually around 0.5. So I was looking for those pairs of traits that on average are correlated at 0.5 or more with the Pearson correlation coefficient. Okay, you can ask me more questions about that later. I know this is not necessarily easy to follow. So let me just show you the results now. So this is what we found. There's like a lot of information to unpack in this graph. So I'm gonna just focus on the two main points that are most important for this talk. So the first main point is that I was looking for those curves that are on the far end of my graph. So I actually found 6 pairs of traits that were on average significantly correlated within my species. Only one of them is in red, the others are in black. And so these are the pairs of traits. These are the average correlation coefficients. I put a little d to those that I also put in black, and that's because those traits are dimensionally related. And what I mean by that is that I have these two traits that I'm correlating, but there's something about this trait, a way that I can express it mathematically that can also be found in that other trait. So maybe the correlation that I see is partly driven by a basic mathematical relationship, or like they're sharing some dimension. So the easiest example I can give of that, so Ks is conductivity per sapwood area, K branch is conductivity per branch area. So here these two traits share a common numerator. So of course we'd expect them to have a minimum level of correlation. So to be really conservative, I'm only going to interpret biologically or ecologically those traits that are not dimensionally related whatsoever. So this is that pair of trait that's in red. So that's carbon-13 isotope ratio and leaf mass per area. So here to show that a little bit more in a more intuitive diagram, this is just a bivariate plot. Carbon-13 isotope ratio is usually understood to be an index of water use efficiency of the whole plant. And leaf mass per area is often associated with the leaf economic spectrum. And here in blue, I have the average interspecific correlation between my species, of my 24 species. And in black is the intraspecific correlation for every one of my 24 species. And what's solid is significant and what's dashed is not significant. So here I only have 3 of my species whose intraspecific correlation are not significant. But if you look on overall, even when they're not significant, like all the correlations, intra and interspecific, are very, very similar in their slope. So yeah, so basically, what we found, I think it's extremely interesting that yes, we did find a fundamental trait-trait relationship that occurs within almost all of my species, but it's not from a trait dimension. It actually, so it's not a leaf economic spectrum relationship, it's not a wood spectrum relationship or an architectural relationship, it's the relationship between leaf structure and water use, which I think is, any plant physiologist will maybe think that's really obvious, but it's really interesting that those trait dimensions we've been thinking about have been largely ignoring the role of water use in defining leaf form and function and leaf carbon acquisition strategy. Okay, oh yeah, let me take you back to the last slide. I told you there were two interesting points in that figure. The other point that I thought was so interesting is that most of the curves, most of those distribution of correlation coefficient for each pair of traits have very large tails. So actually, I don't remember exactly, but in my data set, there were, I think, 20 to 24 pairs of traits which had significantly positive correlation in some species and significantly negative correlation in other species. And then, of course, a bunch of non-significant correlation. So this really illustrates the fact that trait correlations can be very variable among the different species. So, okay, so here's another way to visualize this. So these are other trait correlation networks. Again, color-coded. Here are my trait acronyms. And the different codes here are the species names. So Acer pennsylvanicum, Acer saccharum, Acer rubrum, Acer spicatum. So these are 4 Acer species, 4 maples. Here we have 3 Betula, Betula leganiensis, Papyrifera, Populifolia, so 3 birches. And if you look within a family, like, okay, these correlation networks are very different visually, and even within individual families, they're actually quite different. So that's the other really interesting aspect of that data set. So now the question becomes, okay, can we explain those differences if it doesn't seem to be driven by phylogeny? So, okay, now I have to quantify differences. So how different are each of my species pair? So to do that, I took like a broad approach. So I compared the overall correlation matrix within each species. So remember, I have my trait correlation matrices for species A, for species B. And then I just do a simple mental correlation test, which is this R here, correlation with subscript M. Mental correlation is just like a Pearson correlation. It gives you a value ranging from -1 to +1. So they're easy to interpret. Okay, so this is my response variable. I'm trying to predict how similar are my different species in their intraspecific correlations. So here's basically doing all these mental tests. I have 24 species. Again, I have a bunch of different values. And I can see here's my Aceraceae, maples, Betulaceae, birches, Salicaceae, Rosaceae per family. I grouped them by family. And I color-coded it so that the non-significant correlations are paler and the color darkens as the correlations are stronger. So you can just look, and visually you can also see that there's, sure, like Salicaceae tend to be more similar to each other, but I also have a big cluster here of species that are more similar to each other that are not related. And yeah, here the Rosaceae are not very very similar in their integration structure. But this table now become my response variable. Can I predict those differences among species in trait integration similarity? So that's why I wrote this little y here. It's my response variable that I'm going to try to predict. So, okay, so what explains those differences? So the first thing I thought was, okay, is probably due to environmental gradients. So let's quantify the differences in environment where these different species live. So I quantified environmental variables to assess their similarity. So in that study, I also measured 21 environmental variables, both above and below ground, both biotic and abiotic. Thankfully, I was able to successfully summarize them in a PCA, because that's like a whole lot of variables to work with in addition to all the traits. And it really, came out of the PCA in a way that was very interpretable in terms of biology and environment. So all the variables falling along PC1 indicated soil fertility. Then we had a gradient in light intensity. The third gradient was more like soil water availability. And it was really interesting because soil humidity was correlated with soil depth, so which I think of as reflecting the amount of water that soil can hold. And then the fourth PCA was pH, and then the other two are air temperature. PC5 is air temperature. I had different temperature variables from these I buttons. And then the sixth one is the only one that I can't really make sense of biologically because it combines slope and soil phosphorus. In the future analyses, though, that's like too many principal components to consider given the power of my data set, so I really work with the first four. Okay, so now that I have all these environmental variables, what I want to do is quantify how similar is the environmental niche of my different species. So I did that in two different ways. So this is like in multivariate space. This is, for example, my three principal components, environmental principal components. I located in this multivariate space the individual of that species, and then I created a convex hull, like wrapping those, the volume of the environmental volume where that species occur. And then I did that for each of my 24 species, so I had volumes, and then I measured the distance between the centroids of these volumes. So the distance between, I call them species niche or environmental niche. Okay, something else, another way to quantify niche similarity is to look at the overlap between those volumes, because I can, if I just look at the distance, I could have two volumes that are at a certain distance, but if they're very small, they don't overlap at all. But or if they're very large, then they might actually share a lot of that environmental niche. So my second measure of environmental niche similarity is just niche overlap in hypervolume. And then I also wanted to test more formally whether there was a phylogenetic signal. So between my pairs of species, I reconstructed the phylogenetic tree, measures phylogenetic distance as the sum of the branch length between two species. And so these basically, now I have X, so these are my predictor variables I'm going to use to predict, to test whether they can predict whether my trait integration of my different species is more or less similar. So again, just to kind of go over everything, these are a lot of like analyses on top of analyses on top of analyses. So to summarize, I would expect that as phylogenetic distance increases, the trait integration similarity of my species pair should decrease. Then I would expect that as my environmental niche distance, as measured by the centroid, increases, the similarity should decrease. And on the contrary, as the environmental niche overlap increases, the integration similarity should also increase. Okay, so that's the expectation. What did I find? Results. Ta-da! Drum roll. Okay, no relationship whatsoever with phylogenetic distance, even if you squint and tilt your head. No relationship between environmental niche overlap and integration similarity either. And how about the third one? No relationship either. So all that work to find nothing. So this is actually very puzzling, but I have three kind of thoughts I want to share, which I think are of interest. The first one is that we find that those results tell us that the integration similarity is not associated with the environmental niche as defined within their climatic niche, because all these species co-occur in Mont-Saint-Hilaire, so these populations have the same climatic experience, adaptation. So the fact that there's no relationship between integration similarity and environmental niche based on those variables doesn't mean that that the environmental niche is not responsible at all. It could be that if there's aspects of the environmental niche that occur at larger scale, so it's just as the climatic niche, that maybe these are the variables that explain differences in trait integration, but I wasn't able to measure them because I was working within a climate. So that's the first point. The second point is that even though this is very, I think it's surprising, this is actually consistent with other studies. So as far as I know, I'm the only studies that have looked at plant traits and like physiological traits as opposed to reproductive traits. But again, in the evolutionary biology literature where I got inspired for these methods from, all the studies that have attempted to explain differences in multivariate trait integration, whether they test phylogeny, environmental gradient, or anything, they actually always find like that they can't explain those differences in integration. which takes me, so it's not unique, but that takes me to my third point. These results also make me wonder whether if there is a little signal maybe or something that can be explained by environmental differences, by looking at all my traits together, again, I could be drowning the signal in the noise. I did explore that aspect of my data, and I didn't find anything there again. So I did look at bivariate correlations. I haven't done it, but I've been meaning to look at individual principal components, environmental principal components. And I also looked at so remember I found 6 pairs of traits that were significantly correlated all the time in all my species. So I just did a subset of my data matrix, of my trait matrix, with only these pairs of traits and redid those methods on that, because I was like, maybe to find a signal, I have to work with traits that are actually significantly correlated. If you have correlation coefficients of 0, maybe you just can't find anything that explains 0 because there is nothing to explain. And I also didn't find anything there. So anyways, there's something about the multivariate nature of those analyses that I think might be mean that we might have to think about those questions either slightly differently or use different methods, or I don't know. Again, I've tried to explore that aspect of the data, didn't find anything, but I don't think it rules out that explanation. Okay, so no, species differences are not explained by the environment or phylogeny, as far as I can tell. Okay, so as an answer to the second question, we did find that there's a consistent leaf mass per area and water use relationship amid widely variable intraspecific integration in the different species. And so I think this is a more interesting result, again, because it is a result. And the implication of this is that water use is probably very central to functional design of plants. And unfortunately, it hasn't been incorporated yet in our description of plant strategies. And also the other implication is that it also means that trait dimensions are not universal relationships. So again, they might be global, but it doesn't mean that it's there all the time. Okay, third and last question. So this one is almost, maybe it's the one I'm most excited about. I don't want to have a favorite child here, but I really like that question. So can traits predict plant performance? Or basically, I'm testing the assumption that the traits that we're usually measuring are the ones that actually strongly affect performance. So again, same data set, Mont-Saint-Hilaire, for that first study. Unfortunately, at the last summer of fieldwork, we had to kill all our trees. So this is a picture of Emilie, like after we spent 2 weeks hard at work, just sawing down 380 trees with a little handsaw. So for each of my trees, I have 17 traits. I have 6 environmental gradients, so 6 principal components, remember. And I also now have relative growth rate, which I got as basal area increment for each of those cookies that I measured that I cut at the base. So here I measured relative growth rate as the annual area increment divided by its original size and then by annual per year. And then just as a broad overview, I try to predict relative growth rate as a function of the traits, as a function of the environmental principal components, as a function of species identity, and as a function of age, which I had as a covariate because I could count the tree rings on that tree now. Just as a fun aside, like age, my trees, they all varied. I think the youngest one was 3-year-old, surprisingly, to I think 52-year-old. So yeah, that was nice to see. Okay, so I would expect that the classes of variables that would predict performance, which I measured as relative growth rate, would be, of course, traits, because we assume that traits predict performance. Environment, because we expect that some environments are generally favorable, so more light, usually all plants like more light. And then we'd expect an interaction, a significant interaction between traits and environment, because we also expect that some traits are adaptive in certain environments or not other. Yeah. So what did I find? So I'm showing you my result as a Venn diagram explaining the percent variance explained by the different categories of variable. So from this, I'm going to walk you through this diagram slowly, but first I want to go through the good news. Together, all this model, including the best model, actually predicted 80% of the variance in relative growth rate, which as far as I know, and is really high, and so I'm really kind of surprised by that high number. And the other good news is that traits, so first order effect of traits and second order effect of traits, so the effect of trait, one trait value given another trait value, together they predict 53% of the variance. So that's that number here. So those results are good news in the sense that it supports the assumption that the traits that we measure really affect plant performance. And here you see the covariances, and yeah. The bad news in that model is that in order to get that 53% from traits, we needed all 16 of the 17 traits, and we needed 16 trait-by-trait interactions. So it's not a simple model, and I can't just measure 3 traits to predict plant performance. I need all 16 traits and their interactions. So it's really supporting this paradigm where all the traits and their interaction are needed to understand performance. So that's not good news for trait-based ecologists, because now I can't just go measure wood density and SLA and predict individual tree performance. I need to measure a whole lot of things. So it's definitely not a shortcut to doing ecology. The other bad news is that in order to get that 80% predictive power, in my model, I had individual level trait values. So I tried just for fun substituting my individual level trait values with species mean trait values, and my variance predicted by traits dropped from 53% to 2%. So if I use species mean trait values, I cannot predict individual performance or relative growth rate. So that's the bad news. So can traits predict individual level trait performance? Well, yes, but if you measure many traits and if you measure them on individuals. So it's not a shortcut. And last, I asked that same question. So this is the last bit, and then I'm going to wrap up. I'm aware I'm at 55 minutes. Sorry about that. So I asked the same question. This is my postdoc work I did with Mark Bellin in Sherbrooke, and we wanted to look at whether we can predict population level performance based on traits. So in Sherbrooke, we worked on Montmégantic. We happened to have species survey data in 50 plots along the elevational gradient, and they were surveyed both in 1970 and 2012. And over that time period, the region has warmed up by an average of 1.5 degrees Celsius. So we have an spatial environmental gradient and a temporal environmental gradient. And here they had found that based on basically presence, they looked at the distribution of different species, and they found that the mean elevation between 1970 and the mean elevation 2012 of those different species had shifted on average 36 meters. So the species have been moving uphill to try to fit their climatic niche, supposedly, probably. So the average is 36 meters, but then the question that remains from that kind of result is like, okay, there's a lot of variation about that 36 meter shift in elevation. So actually, can we predict which species have increased a lot in elevation, which species have only increased a little bit, and which species have decreased? And so of course I come with my trait-based approach tools, and I'm like, maybe we can measure traits to say, are there some traits related to their response to global warming that could predict their elevational shift? So in 2016-17, we went on the mountain and we measured the 50 traits of the 50 most abundant understory species. So I'm not working with tree species anymore. And we measured eight traits. So leaf dry matter content, leaf thickness, leaf area, plant height, rooting depth, mycorrhizal fraction, specific root length, and leaf mass fraction. I don't think I put it here, but I think we had 8 to 10 individual per species. Yep. So, and then we predicted actually both shift and elevation, which is what this graph is showing you, but we also had indices of their relative abundance. So we also predicted shift in their relative change in abundance across the whole mountain. So it's an average abundance on the whole mountain across 50 plots. So we predicted whether traits could predict, we tested whether traits could predict that shift in abundance and that shift in elevation. So we found actually that we can predict shifts in elevation in species about, like, these are values more normal to ecologists. R squared is 40%. And actually, so we use different methods. We use regression trees and linear regressions, LMs or GLMs. And so the 2 traits that predicted shift in elevation are leaf mass fraction and rooting depth. And the trait that predicted, there was one trait that predicted shift in abundance, and very interestingly, it's mycorrhizal fraction. So that's like a biotic interaction trait. It's not so much an environmental, it's not so much well, it's a physiological trait, but it has to do with the symbiosis between mycorrhizae and roots. And what's, for me, what I think is the kind of overarching interesting result from that is that, again, none of those traits that matter for the species' response to climate change are traits that we would normally ascribe to one of the trait dimensions. So leaf mass fraction, not really. Rooting depth, well, everybody knows it's important, but we usually don't measure it because it's hard. Mycorrhizal fraction, very few people study it as far as I know. But these are the things that seem to matter. So we did find that some traits, and largely root traits actually, can predict species response to warming, but they're not the ones that are commonly measured. So in summary for that question, we found that individual performance individual performance is predicted by many interacting traits measured at the individual level. And for my population level study, population performance is predicted by seldomly measured traits. So the implication of that is that predicting performance is definitely possible, but from traits, but it's not easy and it's not a shortcut. So in summary, from my 3 questions, we found that global trait dimensions may not hold at local scales. Then we looked at trait dimensions, and we found that coexisting species show very different intra-population trait integration. And the population, in terms of performance response, I'm not going to repeat that because I just said it. So the take-home message from all of this, I believe, is that in terms of phenotypic complexity, at local scale, we may not be able to simplify the phenotype to just a handful of traits. And that's because traits don't follow along trait dimensions, at least in some studies, or in my study, it didn't seem to follow along independent trait dimensions. And also because in order to measure performance, I needed many traits, not just 1 trait or 2 or 3. In terms of scale, we found that trait relationship may not hold across scale, or we just can't assume that they definitely do, because again, trait correlation that are found at global scales are not found locally. So definitely traits are not necessarily a shortcut if you're interested in local scale ecology. It does not take away from the importance of traits in our ability to compare species from different biomes and more global scale comparison. But since a lot of the ecology that we do is at global scale, it just doesn't necessarily translate as a useful tool to use for local ecologists. So trait-based ecology, I'm going to argue where I think trait-based ecology should go. Like, I don't think we should throw out the baby with the bathwater here. Definitely phenotypes are important. This is why we do ecology. But I think we should shift the way we think about trait-based ecology. So if you remember, I started the talk here by saying trait-based ecology currently is around here in that diagram. I think we need to move it over here. So we need to measure more traits. Like, all traits is clearly impossible, but we need to measure more traits. And we need to be very cognizant of how these trait relationships shift across different biological scale, because the traits respond to different drivers of variation that act predominantly at different ecological scales. So if we do that, then I think we'd have a tool and a framework that's much more not plastic, but flexible and applicable to all sorts of ecological contexts. So the next frontiers are to embrace that phenotypic complexity and that scale dependence. Like, challenges are also opportunities. We need to climb that wall. Just go back to how Christian introduced me. Climbing is fun, so this is fun. We definitely should do it, because basically, ultimately, the phenotype is what matters for both ecology and evolution. So I'm not saying we should ignore the phenotype or go back to an autoecology species identity approach. And we can do it, embrace that complexity and scale dependence, because people are increasingly compiling data set. There's increasing sharing of data. So for example, Compadre is a matrix, data set that's been compiled by Rob Salvaro Gomez in Germany, I think he's right now. And he's compiling data sets that have performance metrics with trait metrics. Fred, Fine Root Ecology dataset is led by Luke McCormack and Colleen Iverson, and they're putting together all those root traits that really matter but that are hard to measure. And then there's, of course, TRI, which I've contributed to, and then even Isabelle Aubin is putting together traits of plants in Canada to pick. So we have the data. We also have the analytical tool. So I keep telling people to go read evolutionary biology, because I think they have tons of insight. They're basically asking the exact same question as I am in terms of variation integration, but they've been thinking about it in terms of genes instead of trait. but for at least 50, 60 years. So they've developed like analytical methods that are very powerful. There's like, here's an example of also Bill Shipley proposed to use path analysis as a better way to extract information from correlations. Ben Blunder has developed this hypervolume package to use in R, to use with multivariate data, which is how I got these distances and overlap in environmental niche centroids and volumes. So yeah, we have the know-how, and we have the theory, and we have the literature. So yeah, I think it's possible. We just need to kind of shift our emphasis a little bit. And I think that's it. I want to thank my collaborators, mentors, colleagues, many undergraduates that have done tons of work. It's so many hours measuring these traits on so many individuals, and in the field work, doing very hard field work. And I need to thank my funding agency, so NSERC. I got this Chateaubriand Fellowship, so that's from the French Republic in the US. The ERC that's supported Cyril Viol, who supported me, and then FQRNT, of course, who supported also my fieldwork. So with that, I will be happy to take questions if we have time.
Yes, we will sure. On va prendre du temps, c'est certain.
Oui.
Pour quelques questions, je suis certain qu'on en a beaucoup. Avant que les gens, j'invite les gens à lever la main là, sur Zoom s'il y a des questions. Mais avant de dire, d'abord, merci beaucoup, Julie. J'ai été un peu surpris par le fait qu'une Messier fasse une présentation sans parler des Canadiens de Montréal. Je ne pensais pas que c'était possible.
C'était-tu rafraîchissant ou quoi?
J'ai jamais vu une présentation de Christian qui ne parle pas des Canadiens.
Il va falloir que j'insère dans mes remerciements les Canadiens de Montréal pour garder mon support moral.
C'est ça. Bon, Christian a la première question. Ça va être une facile, j'imagine.
Ben merci, Julie. C'est toujours intéressant. Ça m'a ramené un peu en arrière quand j'avais été écouter ta soutenance de doctorat. Et en fait, ton travail est excellent. Et c'est extraordinaire, mais je me souviens que j'avais sorti ta soutenance un peu déprimée sur le futur des traits. Et j'ai un peu la même réaction aujourd'hui. Ma première question, en fait, c'est qu'est-ce que ça donne de prendre une moyenne d'espèces quand on veut mesurer les traits comme on fait tous? D'ailleurs, il y a des banques de données là-dessus. Est-ce que c'est utile seulement à des échelles plus globales ou... Parce que tu sembles dire que quand tu fais des comparaisons, peut-être à grande échelle, peut-être que ces traits-là, ça peut être utile, mais quand tu fais des comparaisons plus locales, il faut beaucoup de traits et peut-être qu'il faut mesurer les traits de l'espèce ou de l'individu. Donc, j'aimerais que tu me dises quelques mots sur peut-être les limites d'utiliser cette moyenne de traits-là, comme on fait tous, peut-être, et qu'on abuse.
Oui, bien c'est ça. C'est sûr que les gens utilisent des moyennes parce que c'est pas réaliste de dire je vais aller sur chaque individu, je vais aller mesurer 18 traits, là, ça n'a pas de sens. Donc, c'est plus une limitation logistique qu'une limitation. Je suis tout à fait consciente, je ne dis pas que les gens ne sont pas conscients du fait qu'il y a de la variation intraspécifique, ils le savent. C'est juste qu'à un moment donné, il faut prendre des mesures avec les ressources qu'on a, avec le temps qu'on a, puis c'est tout. Je pense que c'est juste qu'il va falloir recentrer ou réfléchir à quelles questions est-ce qu'on peut répondre avec ce genre. Si on est obligé de prendre des moyennes, bon, ben quelles questions est-ce que je peux raisonnablement m'attendre à répondre avec ces moyennes-là? Je pense que mes travaux, mon premier papier qui était très cité, c'est en 2010, qui regardait la quantité de variation intraspécifique au Panama, puis qui trouvait qu'il y avait en fait 50% de la variation totale des traits se trouvait en intraspécifique. Il y a d'autres études qui ont été faites dans avec le même genre de questions. Puis, le chiffre que j'ai vu depuis les 10 dernières années, c'est que les gens sont plus conscients du fait que je ne peux peut-être pas aller prendre une mesure d'un trait, d'une espèce sur Try, puis l'appliquer dans mon site. Fait au moins, si je peux considérer la variation intraspécifique en termes du fait que différentes populations locales sont adaptées à différents environnements, puis au moins aller mesurer mes traits de façon locale, c'est déjà beaucoup mieux. Aussi, évidemment, il y a un autre aspect de mes études, c'est que je voulais aller prédire la performance de chaque arbre. Je ne sais pas si s'il y a beaucoup de questions qui disent « moi, je veux pouvoir prédire quel individu va pousser », souvent, on est plus intéressé au niveau de la population. Une autre analyse que je pourrais faire sur la base de données, que je n'ai pas encore fait, mais c'est de dire « est-ce que je suis capable de prédire le taux de croissance moyen de ma population en fonction des traits moyens de cette population-là? » Puis à ce moment-là, peut-être que j'aurai des résultats plus... que mes moyennes d'espèces fonctionneraient mieux. Mais c'était juste comme je dis, moi, mes études, c'est pas nécessairement pour... C'est clairement pas, comme je dis, faisable d'aller faire ce que j'ai fait là tout le temps. Mais le but de l'exercice, c'était justement d'aller voir quand est-ce que ça marche pas pour pouvoir dire, OK, si ça marche pas là, on va arrêter de dire je vais mesurer SLA sur un arbre puis prédire la performance de cet arbre-là. Puis il va falloir que je réfléchisse de dire, OK, ces outils-là sont bons pour ici, mais après ça, ça l'arrête. Puis en fait, je vois beaucoup de travaux qui ont été faits, c'est ça, depuis les 5-10 dernières années qui sont publiés. Souvent, les gens vont vraiment mesurer leurs traits localement ou ils vont s'assurer d'avoir un gradient environnemental très large pour qu'il y ait un turnover des valeurs de traits, un turnover des espèces. Fait que je pense que ces nuances-là, en termes de complexité puis en termes d'échelle, les gens commencent à plus l'incorporer. C'est juste qu'au début, avec le Leaf Economic Spectrum de Wright en 2004, tout le monde était tellement content. Oh mon Dieu, on a trouvé le silver bullet, on a trouvé on va être capable de tout caractériser en mesurant SLA. Tu sais, mais là, non.
Merci.
Est-ce qu'on a d'autres questions?
Je sais que je rencontre des étudiants ensuite. Je ne sais pas s'il y avait des gens qui avaient des questions, mais qui ne seront pas capables de nous joindre à 2h15, je crois, 2 heures qu'on se rencontre.
Je laisse encore quelques instants. Moi, je vais être avec toi. Je vais avoir des questions pour toi, mais j'aimerais ça s'il y en a d'autres qui
Moi, j'en ai d'autres, mais je ne veux pas monopoliser le débat. Mais...
On peut y aller. Je ne vois pas d'autres mains levées. Donc, vas-y, Christian.
La chose que j'ai trouvée intéressante, Julie, c'est les racines. Moi, je m'intéresse aux racines depuis très longtemps. Et ce que tu trouvais, je pense, c'est super intéressant. On a toujours mesuré, essayé de tout expliquer en mesurant des traits au-dessus du sol. Mais les racines, c'est important. Il faut le répéter. Merci, tu l'as répété. Mais c'est pas facile d'aller chercher ces traits-là. Il nous en manque. Je ne connaissais pas le Fred, là, la base de données Fred, que j'aimerais ça aller voir. Moi, j'en ai beaucoup de données de racines. Peut-être que je pourrais contribuer.
Ah, il serait très heureux de t'avoir.
Donc, je pense que c'est intéressant. Moi, je crois beaucoup que l'avenir est beaucoup à intégrer les racines et évidemment ce qui se passe au-dessus du sol.
Puis,
je pense que ça va nous aider énormément.
Puis Luke McCormack et Colin Averson sont super fins. C'est des gens très agréables pour travailler.
Je connais McCormack, je ne connais pas l'autre, mais OK. Ouais.
Puis pour revenir sur cette histoire-là, évidemment, la réponse de l'espèce au changement climatique, c'est l'espèce, leur niche, c'est multivarié. Fait qu'en fait, je n'ai pas eu le temps d'expliquer, mais je crois que ce qui se passe, c'est que même si leur niche climatique monte en élévation, sur le mont Saint-Hilaire, en fait, on passe à la base d'une forêt mixte à une forêt boréale. Donc, le sol est très différent. Fait que ça se peut que leur capacité de migrer vers le nord soit limitée parce que, tout à coup, dans un sol boréal qui se décompose peu, qui est très acide, ben, l'espèce, elle, ce qui est adapté, c'est une combinaison de tel climat avec tel sol. Puis, avec les changements climatiques, on va avoir du découplage intense des axes de niche des différentes espèces. Fait que ça se peut qu'il y ait bien des espèces qui ne soient pas capables de monter, de suivre le climat parce qu'il y a d'autres variables environnementales qui ne suivent pas.
Si vous permettez, j'avais une petite dernière question. Là, ton analyse de lien avec les traits en utilisant, si je comprends bien, une analyse de réseau, en fait.
Oui.
Est-ce qu'on pourrait même regarder des traits qui seraient des nœuds plus centraux, qui auraient plus d'importance, plus liés aux autres?
Oui, absolument.
Et, faire une analyse de ce type-là, c'est quelque chose qui m'intéresserait personnellement. Je pense qu'il y a Nuria qui écoutait ça, qui fait ça énormément.
Oui.
Il y avait quelque chose d'intéressant à faire.
Dans mes analyses, je ne l'ai pas présenté, mais dans ce chapitre-là, c'est mon article, je crois, qui est dans Journal of Ecology, j'ai fait cette analyse-là de centralité. Puis le trait, je ne me rappelle plus par cœur, mais c'était un trait feuillage, je pense que c'était Leaf Area. C'était très intéressant parce que, en fait, là, le débat, c'est OK, est-ce que ça contient plus d'informations ou moins d'informations, le trait, le fait qu'il soit central?
Ouais.
S'il est central, ça veut dire qu'il est affecté partout, mais s'il est affecté partout, quand il y a de la variation, on n'est pas capable d'interpréter.
Je peux rien dire. Ouais.
Ben, donc, est-ce qu'il y a une façon d'utiliser de façon intelligente, de dire, ben, si c'est corrélé avec tout, je fais quoi avec, en fait? Puis en fait, j'avais fait des tests de voir est-ce qu'on est capable d'isoler c'est quoi les modules qui émergent de mon réseau de corrélation. Puis on en a trouvé 3. Mais c'est des affaires que moi, Marty Lekowitz, Brian McGill, puis Brian Enquist, on a passé du temps à regarder, puis on n'a aucune idée de ce que ça veut dire. Parce qu'au lieu d'arriver avec des suppositions a priori, de dire je devrais voir ces trait dimensions-là, peut-être qu'on dirait ben, je vais laisser les données me parler, puis je vais voir c'est quoi les modules qui émergent.
Qu'est-ce qui
Puis
émerge?
c'est des affaires que je ne comprends pas.
OK. On devrait en rediscuter avec des gens qui font ça à d'autres échelles. OK. Bon, je me tais là. Je ne parle plus.
Okay. Merci beaucoup, Julie. C'était très, très riche comme présentation, très complet. C'était complexe aussi, mais tu as su bien le vulgariser. C'était vraiment intéressant. Un grand, grand merci.
Merci, ça m'a vraiment fait plaisir de vous parler. Puis j'espère avoir l'opportunité de visiter en personne puis de pouvoir m'asseoir avec chacun de vous avec un café ou une bière puis discuter de tout ça. Là.
Ben oui. Et d'autres choses.
Absolument.
C'est ça.
Oui, jouer au hockey.
C'est ça. Pour les étudiants du cours, on se revoit dans 30 minutes à 2h20. On va laisser le temps à Julie de manger un petit peu, puis on se revoit à 2h20.